Well, hello, everybody. I'm Charlie Brummer at UC Davis. Thanks for tuning into the seminar. I'm really looking forward to the discussion we'll have on Friday. I want to thank Jennifer for inviting me up there. And even though I can't be there in person, at least we have the Zoom option these days, and that'll have to do. And I have to say, I'm not missing the cold and snow too much, so I'll be okay. Let me share my screen and we'll get going with the talk here. Okay. Somehow or other, this just came to me out of the blue when I was thinking about getting this talk ready, there and back again. And of course, that made me think then of The Hobbit. And so in thinking about it, I said, let's call this the Thremmatologist, or There and Back Again. And hopefully it'll become clear as I go through why I'm doing that. So thrematology is the science of breeding animals and plants under domestication. And since we're not going to be talking too much about animals, we're mainly focusing here on plant breeding. And the thrematologist, of course, is the plant breeder. So you might be asking, well, what is this talk about? What I hope it is a discussion about plant breeding and about how plant breeding proceeds, illustrated with examples from my research, mainly on alfalfa, but a few other species thrown in as we go through. I want to thank Bill Tracy for a very interesting paper from a few years ago in the Seeds and Breeds Symposium. And some of the things I'm talking about are echoes of things that he brought up in that paper, and then hopefully I'm bringing up some additional things as well. And I hope that you'll find it interesting, or at least worthy of discussion later on. What we'll talk about are sort of an overview of what plant breeding is, some various concepts of plant breeding, and sort of looking at a pipeline of plant breeding from start to finish, discussing a little bit about a plant breeding toolkit and new technologies and their role in the plant breeding pipeline. And then I want to talk about the plant breeding education program that we have called the SCOPE project, where students get practical experience breeding. This picture is of a spinach breeding trial from down in El Centro from just about a year ago, actually. So what is plant breeding? The way I look at it, plant breeding has a direction, a goal, which is the development of a new variety, a new cultivar that somebody is going to find useful. And that sort of sets it as a distinction from other plant science or plant biology research. Plant breeding is really the targeted manipulation of plants to improve or introduce one or more traits that are reliably expressed under production conditions. And I think this reliable expression under production conditions is probably a really critical component of plant breeding. If it doesn't always show the traits we want it to have, it's going to be very difficult when we try to market the product. And again, that goal of a tangible product at the end is what really makes plant breeding a distinct discipline, other and different from some of the other more research-driven disciplines in plant biology. In brief, we can look at breeding like this. You start with germplasm, you end up with a cultivar, and in between, you've done something to evaluate and select, and maybe that's all you did, just evaluated and selected a bunch of different lines and released one as a cultivar. Or maybe you recombined the best lines and tried to develop more variation and went around the recurrent selections in some form, the recurrent selection cycle several times before you had something that was adequate to release as a cultivar. One of the things that Bill says in that paper that I mentioned earlier is the direct effect of selection is remarkably predictable and precise. You get what you select. And so if you want to select pigeons for fantails, as Darwin talks about, then you can do that as long as you can see a fantail, you can select it, and you will get what you want. And so what we can say generally about breeding is that even not knowing anything about the genetic basis of a trait, if there is genetic variation and the trait is heritable, then you'll make selection and you'll get what you want. And kind of a corollary, and this may be obvious, although I don't know that we always think about it as much as we should. If you don't select for a trait, you're unlikely to see gain in that trait. And so if you're doing something that isn't going in the direction you really want, well, maybe you need to redo the way you're doing selection so that you can get what you actually want. You get what you select for. I like to think of breeding in terms of a pipeline, really, where you start with germplasm and then you go through this process of developing populations, or you're creating families. In an inbred species, you'd be creating inbred lines. You evaluate them in target environments to find the best ones. If you're evaluating lines of some crop, you could directly move them into trials for release. For alfalfa or many forage crops, we then hybridize the best selections to develop an improved population, which we then trial in multi-locations for release. Of course, once we do hybridization, we also generate new recombinants. So there's new variation there that we might want to look at, and we can run back into our evaluation nurseries. So there and back again, we developed something, it looked good, we recombined, and we're back to the beginning to go through again. And then we can continue to do this cycle and churn out new varieties at the other end. Now, something that's really important in terms of plant breeding and in terms of making sure the products of plant breeding actually go somewhere besides just a journal article is seed production, or in the case of trees, for example, maybe it's budwood production, clonal production. The seed stock needed to get out to the ultimate user of the material, in this case, a farmer or a rancher. So during this period of evaluation, there's a concurrent increase of seed so that seed's available when a cultivar is ultimately released, and you don't wait several years to produce seed once you have a cultivar that's worthy of being commercialized. Now, before we really get into that whole scheme of a breeding program, as a breeder, we need to think about, what are we doing here? What is our cultivar concept, if we want to look at it like that? What are the trait combinations we need to have? What are the threshold values we must meet? What is our ultimate goal? And once we know that, then we can make sure we're looking at the right germplasm, we're using the correct breeding methods, and that might be recurrent selection, it might be back crossing, it might be any number of different ways to get at the ultimate variety. But we don't really know what the best way is unless we really have a clear goal in mind. And once we have that goal, also, we can develop a timeline to figure out how soon it is that we can develop something, get all the gear, all the pieces in motion or in place. So once the gears get in motion, we can move through the system in a sensible way. This isn't always easy to do. And in fact, I've gone down this road myself many times without having a clear conception of where we're going, and you end up in the middle and you're kind of muddled as to exactly what to do next. The germplasm base that you have is very important, of course, in terms of making progress. And it's important to understand where the germplasm comes from. Michael Russell, several years ago, a couple of decades ago, wrote a very nice paper about the origins of alfalfa and various other aspects of alfalfa. And he talked about the introduction of these historical germplasms into the United States. Many of these bulletins, or these germplasms, were written up in bulletins over 100 years ago. And there's a lot of good information in these bulletins. And so I'd urge all the students, especially, everyone really, that, if you're interested in your crop, go back to these really old articles and figure out what folks initially saw, what kind of germplasm they were originally obtaining and putting into cultivar development programs, because you can learn a lot from what happened in the past, and we don't end up having to reinvent ourselves over and over all the time. So these are very interesting papers about alfalfa germplasm. A lot of times, you talk to students about genetic diversity, and everyone says, "Oh, genetic diversity is a great thing. It's a really great thing," and it's true that genetic diversity is a good thing. But it's not necessarily a good thing in all places. And so if we look at a sort of a series of different, let's look at these as breeding programs, where A is the germplasm collection at NPGS, where we have a really broad amount of germ diversity in that collection, probably not assembled in a way that's useful for a cultivar. And then we have the improved land races, early cultivars, and so on, that form sort of the base of the commercial breeding programs for the crop. And then the elite breeding programs being C, where we have a very much smaller amount of diversity, but better performance. And then, of course, really good performance of a very narrow slice of germplasm in cultivars, and the elite cultivars, which would represent D. And so, we can look at this as yield, or we can really look at it in multidimensional space, and the fact that you have lots of variability in this reservoir of A that you don't want in a commercial breeding program. On the other hand, other than for single genes and so on, where it's easy to pull genes out and marker-assisted backcross them into a new cultivar, it's hard to get the corpus of that germplasm collection into a state that's useful for breeding. And undoubtedly, there are good alleles in all that kind of messy germplasm. So how do you do that? Move really from A to B. So we have some more germplasm, hopefully carrying different alleles for some of the different environments we might be facing in the future, different stresses. We need adaptation in different ways as we move the crops to different environments and so on. And so we've started an alfalfa pre-breeding project to get at that issue, moving from A to B, where you take germplasm that's not particularly useful and put it into a useful package. This germplasm enhancement project has been funded through a couple grants from the NIFA alfalfa forage and the alfalfa and forage research program. And it includes collaborators in Madison, that's Heathcliff Riday, at Cornell, Don Vines and Julie Hansen, Brian Iris, the curator of the alfalfa collection in Prosser, Washington, and recently Esteban Rios in Florida has joined into this project. The idea is to take this germplasm, do kind of quick recurrent selection, phenotypic recurrent selection for adaptation to different environments, and then to develop geographically distinct or pools of germplasm from geographically distinct origins for the northern or the more dormant germplasm areas, and for the southern or west coast, less dormant germplasm regions. And in this way, have populations that could potentially be commercially useful down the road from germplasm that initially wasn't useful in a commercial sense. And so here are several trials that I've had of NPGS germplasm, ranging from Tule Lake up on the Oregon border to El Centro down on the Mexican border, and 5 Points, which is down in Fresno County in the San Joaquin Valley. The 5 Points was under salinity stress. The El Centro one was under drought and heat stress, and Tule Lake was under cold stress. So looking at these different environments, trying to find which of this germplasm might be useful for future breeding, and putting together germplasm populations. Thinking about developing a breeding population in a crop that you're just starting out in, and in this case, I'm showing, this is the only slide I'll show of hemp, of a hemp breeding project that we started a couple of years ago, trying to get high cannabinoids or high CBD concentration in a package that's a machine harvestable, high density planting adapted cultivar. So what we're really trying to do at this point is to get this single stem architecture phenotype merged with a short stature day neutral flowering phenotype with, of course, large colas, or that central inflorescence that has high, or at least reasonably high cannabinoid concentrations. So we're not really doing any at this point, any sort of recurrent selection type thing. We're simply making crosses among these germplasms to accumulate these different characteristics into the same background, from which we can then focus on population improvement. Now, the foundation of a breeding program and of the germplasm of a cultivar, really, that's commercially relevant, is quantitative traits such as yield and adaptation and potentially quality. Quantitative traits where we have a distribution of phenotypes in a population. And for alfalfa and many of the forage crops I work on, we're talking about outcrossing populations. We don't really have inbreds in any way or produce hybrids, or at least single cross hybrids. And so we have this distribution. We select through truncation at the top end, intercross plants, and hope to move the population forward. This is summarized in the breeder's equation that we all know, where genetic gain, or the response to selection, depends on the proportion of plants that we select and the heritability of the trait. And if we have a heritable trait, we're going to make responses, as Bill and as Darwin pointed out earlier. The key points: it's a numbers game. Breeding for these traits is a numbers game. The larger the population that you can reliably evaluate, the better. Higher heritability is better. And so heritability is not a static trait or a trait doesn't have a static heritability. And so you can improve heritability, change the experimental design, change how you're measuring things, improve phenotyping methodology, and so on. And advancing the heritability in that way by minimizing error and so on is going to improve your gain. And of course, time. And so generally in breeding, faster is better. And it really isn't the response to a cycle of selection that matters so much. It's the response per year. And so trying to get that response up on a yearly basis is what much plant breeding research is geared toward. Glenn Burton, well-known Bermuda grass breeder down in Tifton, Georgia, once said that increased yield is the bottom line in most plant breeding programs. And generally that's true. Yield is important. At some level, yield is important. And I'm going to talk about yield in alfalfa in the next few slides. When he says most, what he's meaning is that some things like lawn grasses, turf grasses, you don't want to have high yield. This is a picture from when I lived down in Georgia. Zoysia grass lawn, I mowed maybe 2 or 3 times. Yield is the last thing I want in that yard. So there are certainly cases where yield's not important, but generally it is. And generally we see in the main oilseed and staple crops that yields have improved. These are USDA data for the US. Yields have improved over the last century in an almost linear fashion in all these cases, as well as for maize silage, where the whole plant is harvested. Interestingly, though, for alfalfa hay, we don't see that same improvement, except for a period from around 1960 to 1980, when the major diseases were improved in alfalfa cultivars. Subsequent to that, we've had other disease problems and pest problems that have been managed, but we really haven't done anything on increasing the yield overall of alfalfa hay. And I think that's a problem in terms of keeping alfalfa in a crop rotation where it's providing a lot of benefits to the system, but it isn't now yielding, and so it's going to be less likely to be included. Okay. The way a lot of alfalfa breeding and forage breeding in general is done is to start out with spaced plant nurseries that is not the production environment for the crop. You can look at large populations of plants this way and get an indication of how individual plants might perform. But there's very low heritability for yield in general, and in many cases, we don't even measure yield on single plants. And at the top there is a picture of a trial down in El Centro where we're looking at germplasm, and you see some accessions that look pretty good and some accessions where most plants have died. So by going through space plant nurseries, picking plants that look good, vigorous, and so on, we might expect the yield to be improved, but we're not actually measuring yield, and it's not in the production environment. And so as Mike Hassler and I wrote in a paper a couple of years ago, we conclude that across the cool season perennial forages, including alfalfa, little to no yield gain has been observed. And in general, these programs have run off of space plant nurseries. The programs that have focused directly on yield using something like Halfsub family selection as a selection criterion have improved yield. And so this has led me to think that we really need to, if we're going to make alfalfa a crop that's going to be useful or potentially competitive with other crops, we've got to improve yield. And so we've got to move to a family-based selection. So that's what we're doing now, looking at Halfsub families, and ideally in solid seeded sward plots like we have in the picture on the left. It's about 80 Halfsub families in a replicated trial that we can measure with the machine harvesting under production conditions. We often don't have enough seed, and that's a big problem for Halfsub sward evaluations. And so in this case, in the middle here, we've transplanted 24 plants in a plot, and these represent kind of, constructed swards and hopefully enough competition among plants that we can somewhat simulate what the real conditions might be. And in some cases, we have very, very little seed, as in the top right, where we have little 8-plant plots of progenies. At least we're looking at progenies and not single individual plants. But nevertheless, this gives some competition, but not clearly what we would like to see as the plots over on the left. But moving to this type of evaluation will hopefully improve our yield. And at the same time, we're using these experiments to test some genomic selection capabilities as well. One of the big problems, of course, with forage crops is that a given experimental unit, so one plot, one Halfsub family and one rep of one location, is going to be harvested somewhere between 4 and 10 times a year, depending on the location, and over several years. Usually we have these trials in the field for maybe 3 to 4, maybe 5 years to get an idea of how the long-term persistence is of the variety, or the family. And so if we could somehow get around this constant harvesting and be able to predict yield using a drone-based sensor, that would be ideal. We could look at much larger populations. We could get a much better handle then on large populations and the yield of large populations, and hopefully make gain from that. And the figure here is an example of doing just that, where we predicted yield based on the measured yield from one harvest, and multi-spec sensor data estimating volume of the canopies of the plots, and then summed up across the harvests and compared the predicted yield with the total yield. And it turns out that the correlation is pretty good. We'll see how that stacks up again. Last year we did it again, and in some of those newer plots as well. This could increase the size of our trials, and as long as we can manage field variation, another problem, but hopefully manageable, then we should be able to influence the gain equation through at least the first part, and hopefully through the second part, through better management of fields and getting closer to the actual trait, rather than having this, on spaced plants, a kind of correlated trait with actual yield. As far as genomic selection for yield goes, we see a lot of promise in using these technologies. We've had this collaboration with Cornell for a number of years with Julie Hansen and Don Vyans, where we had looked at a selection program and developed a predictive model that looked pretty good. We then applied that model to genome or marker only selection, and selected plants using markers both for high and low yield, as well as a random population in the middle, and then evaluated these populations for yield at both Ithaca and in Tule Lake, which is not the target environment. The model was built off of Ithaca data, and even though it's at the same latitude, it's a very different environment, irrigated and so on. That's shown in the picture. Well, the results from 2-year yields were that in Ithaca, we see that selection for high yield is significantly higher than selection for low yield, with a random population in the middle. And that was in the target environment. In the non-target environment, the trend is there, but statistical significance isn't even close. And so this suggests that the markers work and that we can make some potential progress on yield. Now, when you compare the original population with the selected population with markers, these first 2 bars, the orange versus light blue, you can see that there's no difference in yield, although there is a tendency toward higher yield, which is, I mean, at least it's looking a little positive. And compared to phenotypic selection, which took 4 years to do instead of 6 months, we can see we have a higher yield with genomic selection. So that's promising. There's a lot of caveats that I'm not going to go into with this study, but the data show that it looks likely we'll be able to use markers in some way to predict and produce higher yield. This doesn't work in the non-target environment, and so it shows the importance of having a model built for the target environments that you're going to grow the plants in ultimately. Okay, so, well, let me just summarize here and say that between genomic selection, drone-based sensors to increase the size of nurseries, and then using Halfsub families as an actual direct representation of yield rather than using individual spaced plants, we think we can improve our predictive ability for yield, as well as then improve the yield of populations and cultivars that come out. And hopefully we can get that graph back on an upward slope. That remains to be seen, but here's hoping. Now, beyond that kind of foundational type of research, the breeder's equation type research and breeding, we can also think of other breeding objectives that really don't have anything to do with the breeder's equation. They really are just sort of building on that foundation, like introgressing a resistance gene. And here's an example that Travis Parker, who was a student with Paul Gepps, something he did as part of this SCOPE project that I'll mention later that I directed at UC Davis, where he backcrossed bean common mosaic virus from an elite commercial cultivar, that's this white cultivar here, into a variety of different heirlooms with cool coat colors. And in the process of backcrossing, he brought along the virus resistance, recovered the coat colors, and in some cases improved other traits like yield or plant type. And these have resulted in a series of cultivar releases out of this program. These are all public releases from this SCOPE project that was funded by the Organic Research and Education Program. Another thing that you might do once we have a foundation of good traits in place is just look for novel products that can come out of a breeding program. This is another project of the SCOPE project where we were trying to develop a better jalapeno popper. And what we ended up with is a pepper that doesn't really look like a jalapeno, wouldn't sell as a jalapeno or be marketed as one, but it has very many desirable characteristics, as in it has thick walls, it bakes well with cheese in it, has very good flavor, and it's a possible new product. It's not a jalapeno pepper popper, but a new type of pepper popper. And we identified these somewhat tongue-in-cheek with our eyeballs. With just data, we would have thrown this out because it didn't fit a jalapeno model. But looking at it, looks pretty nice. And then we had some on-farm trials with organic farmers who liked it as well. And so it's something that we're going to trial again this year and potentially move toward commercialization. All the different breeding programs can be thought of, collectively as part of this pipeline. And the question is, and it's always raised, especially when talking about new technologies, is breeding slow or fast? Well, everyone asks, is it slow? I say, is it fast? Well, I'm going to talk about wild rice for a minute. I have a little wild rice program that isn't very large. Wild rice is outcrossed and sold as a population. And here's kind of a quick timeline, what we might do: develop progenies, evaluate them the next year and select the best plants, intercross those plants, and then go into variety tests as well as seed increase for potential release. The next year, we would take the best plants also and make families to evaluate and go on through cultivar release and so on. So that in any given year, we're evaluating new families, we're intercrossing the families from the year before, we're doing small plot variety trials from the year before, all of which lead to cultivars. So even though it might take 6 or 7 years to get to a cultivar initially, once we start the pipeline and kind of feed things through it, the pipeline produces varieties every year. And so breeding really isn't that slow. Once it's prime, things come out rapidly. And you can, of course, adapt what traits you're selecting as needed within that whole pipeline process. Nevertheless, anything we can do to speed up the cycle is something that we want to do. And so here, just showing again the breeding pipeline. And then for an alfalfa program, what can we do to speed things up? Well, we can't really do anything about assembling germplasm, although there may be some issues there, of course, with import permits, that kind of stuff. But that's going to always be the case. What can we do about developing populations or developing families or lines? In alfalfa, it will take a season, probably a summer if you want to produce a lot of seed. If you don't need a lot, you could do it in the off season. So that's one way to move things ahead. If you're developing lines in another crop, of course, you could develop doubled haploids or something to move the process along faster and save a little time. We get to the evaluating of plants and so on, and looking at selecting top performers. Could we improve on the time it takes, the 2 to 5 years per cycle in a perennial crop? That really seems like a long time. What could we do about that? Could we improve? And yes, I think it can. Well, maybe we can do it. Here's the rub. We could use genomic prediction. I'm sure that we can do this in some way to make some selections in the off season, maybe make several cycles of selection. I don't know if we'll see how possible that will be before we have to go back out and retrain the model. And so it might be that we can kind of alternate long cycles in the field where we're retraining models, and then short cycles in the greenhouse where we can make quick progress on yield. But we still need to assess other traits. And so it's not really a situation where we can go around and make a couple cycles of yield-based selection based on genomic prediction and forget about other things like long-term persistence or other characteristics of importance. So there's definitely room in there to rejigger the breeding program and cut time off of the overall process. And that's pretty exciting, actually, because up until now, we really haven't had that opportunity. And then you get to the end of the pipeline where we're looking at evaluating and seed production and so on. And it doesn't really seem to me that it's likely we can cut much time out here. So really, we're talking about the meat of the program is either in these family development or in the assessment phases where we can possibly make up for the long selection cycles. Up until the last 5-6 years, maybe, we've done a lot of work, and these are just some of the papers we've published over the years looking at different marker technologies, different mapping approaches. We can map QTLs for yield. We can identify QTLs. We can look at different types of platforms. We developed a SNP array, but for routine breeding use, it's really too expensive. Other markers are too slow. Things were looking kind of problematic. I mean, I developed this map in '93, and 20 years later, we really hadn't done much with markers in alfalfa. And that's largely because it's a population-based autotetraploid outcrosser, and it's hard to work with markers or single QTLs and things. However, when we have genome-wide markers with genotyping by sequencing, with all the problems that technology has, we're finally able to really probably analyze any given population and do things on a population basis that we really couldn't do so well before. And it allows us to get out of this single QTL, either GWAS or biparental population mapping and so on, and get into genomic prediction. And I think that's pretty exciting. And that's really where the future is and the possibilities of doing some cool things in alfalfa. I'm just going to make a very simple comment about transgenes and gene editing. These technologies would be very useful in breeding. They're not going to shave a whole lot of time off the breeding cycle, but they will allow us to introduce new traits potentially, or to expand variation in existing traits that we just don't have. And that would be fantastic. It all depends on regulation and the regulatory environment. And until that changes, it's really just not an issue in a crop that doesn't have that big of a seed market that can cover the costs of all these regulatory demands. And so for alfalfa and other crops like that, there's a lot that can be done with transgenes and gene editing in theory. In practicality, it's driven by regulatory. And that's especially true for export crops like alfalfa, where a considerable amount of hay goes overseas, and it depends on what the other country's tolerance is as to whether you can market it. Okay, I want to switch over and talk about scope now for a few minutes. One of the things that I've always felt at the various places I've been over the years is that our students get a good training in coursework, in plant breeding, genetics, genomics. I mean, there's lots of gaps. Everyone has gaps and we can't cover everything. But nevertheless, theoretical knowledge of breeding and of the new technologies, especially if thesis projects are oriented around those technologies, is generally pretty good. And certainly that's the case at Davis. But the training and practical aspects often is missing, even in programs where a practical breeding program is part of the overall program. Students don't necessarily get involved in the actual hands-on day-to-day activities. So I had this idea a few years ago to start a project where we would have students develop small breeding projects in different vegetables themselves, and kind of run a program themselves with some oversight by faculty. And we called this program SCOPE, for the Student Collaborative Organic Plant Breeding and Education Project. It was funded by the OREI program. We just had it renewed last year. This is a collaborative effort between the Plant Breeding Center, the Student Farm, Cal Poly Pomona, the Organic Seed Alliance, organic farmers in Northern California and elsewhere, and organic seed companies in Northern California and elsewhere as well. And so far, we've had something like over 20 graduate and 70 undergraduate students have been participants since 2015 when we initiated the project. We have everything from on-farm trials. We're showing a couple examples here, field days, seminars and internships, leadership opportunities, all put on by graduate students with undergraduate support. It's been a very exciting program so far. For the new project, we have a number of crops that have, timelines laid out, breeding targets, and so on. And the ultimate goal is to release cultivars. I mentioned the ones released by Travis Parker and Bean earlier in the talk. These came out last year, and one of them will be out this year. The crops that we have going forward represent self-pollinated and cross-pollinated crops, both summer and different seasons as well. The Common Bean project is winding down since we've done what we wanted to do, heirloom varieties with resistance to BCMV. The pepper project I mentioned, we also have a bell pepper project as well. These are moving toward release, hopefully in a couple years. And then we'll make a decision on whether to continue with peppers or not. We have a tomato project, a organic wheat breeding project, and then a new project in zinnia that we just started during the pandemic year, which is interesting, but we did manage to make some crosses, and we're starting to look at progeny in the greenhouse now. So students don't know anything about cross-pollination, it turns out. I'm a cross-pollinated guy, that's all I think about. But many students can't get out of the inbred mindset, and how do you deal with populations. And so I wanted to make sure we had something with population-based, some population-based crop in this new project. Again, these are student-led programs. So it's organized into teams for each species, although a lot of students go between projects as well. The students are in charge of all phases of the program, from developing breeding goals to identifying germplasm. We do marker selection for resistances in the tomato project as well as the bean project. And then doing everything from putting in trials to taking data, analyzing data, making selections, and thinking about the release procedures we're going to use for the different potential cultivars that come out. Now, I should mention there are two staff people, Antonia Palkovic and Laura Roser, who really make sure the teams keep going and that things are done in a sensible way. And really, without their help, the project wouldn't perform as well as it has. Okay, so at this point, I just want to return home from this kind of quick breeding trip. as Bilbo comes back from the mountain. Breeding is a continuum from generation to generation, not only generation of the plants, but generation of breeders. We build on what happened in the past, and hopefully we're leaving a future that can be built upon as well. Programs are built on a germplasm foundation that has adequate yield, quality, and adaptation for our growing conditions. And then we can augment that as needed, as new diseases show up, for example, or as new marketing opportunities arise by novel traits and introgressions or gene edits and so on. Once a pipeline's in place, cultivars come out regularly. And so as long as it isn't disrupted by funding cutoffs and so on, a breeding pipeline is a pretty powerful thing. Every year you're doing you're starting new projects as well as moving other projects along and getting varieties out the door. There's always room for improvement. And Glenn Burton mentioned this in the paper from which that quote came from that I showed you earlier. Constantly trying to improve your ability to assess the phenotype in a way that makes most sense and to move things as fast as possible through the system. And there's various technologies that I mentioned that almost certainly are going to speed things up. And if they don't speed things up necessarily, they'll make us more efficient when we do our selections. And so it was exciting to see how that might work out. Although working on a number of minor crops, what we often see in major crops may not be quite as useful in other crops that have smaller markets and so on. Nevertheless, things that are potentially possible today are very different from what they were back when I got my PhD, longer ago than I'd like to say. Finally, students need training in practical breeding, and I think the SCOPE project that we have does that. So before I wrap up, I want to make a little coda, a story about Don Rasmussen from the Crop Science Society meetings a few years ago. So I guess there may be some people up there who don't know Don as a former barley breeder, but I happen to be sitting behind Don and to the side a little bit at a plenary talk with one of the speakers who's trying to get us motivated to do the next great thing. I don't remember who it was. Don had this notebook on his lap, and I don't think he wrote in it the whole talk, but at the end of the talk, when it was done, and I'll never forget this, he wrote this: "Work harder, do better." Closed it up and off he went. So that's my advice or recommendation to all of you, and me too. Let's work harder, let's do better, let's make better cultivars for the world. So thank you. And I want to thank also the sponsors of my program. I've been very fortunate to be funded by a number of different groups over the years, from competitive grants to commodity organizations, and at various times companies as well. And really it's this mix of funding that makes breeding in a public sector possible, and hopefully it continues. So thank you very much.